right, good afternoon, everyone. We're here today with Kate Smith, who leads our APAC efforts for the early phase division at Worldwide. And Kate is going to be taking us through a presentation called Start Here, Lead Anywhere, the ANZ Advantage in Clinical Development. Kate comes to us with over 30 years of industry experience. And I will turn it over to Kate in just a second. The only housekeeping note that I did want to make mention of is we are going to have Kate go through her presentation, and then we'll have plenty of time at the end for Q&A. So if you do have any questions as Kate goes through the material, you're welcome to drop that in the chat, and then we'll address those and have more of a dialogue once the slides are complete. All right, Kate, over to you.
Great, thanks, Mel. So good morning, good afternoon, everyone. Kate Smith, I'm actually based in New Zealand, so really excited about the presentation today. And as Mel said, there'll be plenty of time at the end for questions. All right, so I'm just going to spend a little bit of time today taking you through some of the key advantages of running clinical trials, particularly early phase trials in Australia and New Zealand. We'll work through timelines, regulatory environment, some of the potential cost savings, and how you can start your development program in ANZ, take advantage of the leapfrog that you get by doing that, and then be able to transition to other regions for the later phases of your development. So on the agenda today, we're going to go through the regulatory and speed efficiencies, cost and incentives, the data and infrastructure, and then also access to patients and the diversity, which is really important for everybody's development program. So here in Australia and New Zealand, we've been keeping a secret for quite a while. This is actually how the world is. So as you can see, New Zealand and Australia, we're actually at the top, and we lead the world in terms of clinical trial development. So just a summary of the key advantages of Australia and New Zealand. So as a New Zealander, much as it pains me, New Zealand and Australia really are like one large country when it comes to the delivery of clinical trials. It's a very, very similar process for both. Same documentation that's required, same level of data that's required to be able to get into clinic. So in New Zealand, we have an approval timeframe of about 4 to 6 weeks. There is the Scott Committee, which provides robust scientific review. It's really important, particularly for those development programs that may want to do something a little bit unique in the early phase. The Scott Committee allows us to have a dialogue and discuss potential different ways of running clinical trials that sometimes is not always available in Australia. No IND requirement, and that's in both Australia and New Zealand, and that's really key, and we'll go through that in more detail. Without taking the cost, the R&D rebates into account, New Zealand and Australia generally are about 30 to 40% cheaper, can be more than that once you take the R&D rebate into account. In New Zealand, we have really high patient retention and adherence. That's primarily due to the fact that a lot of our patients don't have access to key medications because we're a government-funded healthcare system. And we have a really collaborative research environment here, which makes bringing studies from healthy volunteers through to patients really easy. In Australia, very similar startup program. We just have a TGA notification step that's no regulatory review. Total startup days is about 80 days. That takes into account both the startup timeframe, contracting, and also getting all of the other aspects of the trial ready to go. Similarly to New Zealand, significantly cheaper than running a trial in the US, and Australia has a very well-established R&D rebate scheme, which allows rebates up to about 43.5%. There are some hooks to being able to get that rebate, but it is certainly something that a lot of our biotech companies take advantage of. And similar to New Zealand, we've got world-class units here in Australia, and they're very close collaboration with the hospital setting. So this is just showing the difference in the regulatory environment between Australia and New Zealand and the US EU. So as you can see, the ethics timeframe New Zealand and Australia is very short for both. Australia, the sites that we work in, particularly in early phase, tend to use a private ethics committee that meets on a weekly basis. New Zealand has a centralised government-run ethics committee, so the review is very standard and there's a meeting held every week. US and EU also has a short ethics timeframe. However, the total startup timeline is significantly longer due to the fact that you have to take the regulatory application into account. In New Zealand, we don't need an IND or an IDE, we don't need an IMPD, and we need minimal data. in order to get those trials approved. So if you're starting from the same point at the, say, when your preclinical tox is available, general rule of thumb is that we can get into clinic in New Zealand and Australia as much as 4 to 6 months faster than you can in the US. So this is walking through the startup process in Australia. It's a very streamlined process. The 8 weeks gives us sufficient time to get a fully loaded database available as well. We can do a couple of weeks faster if you choose to go with a database and release the edit checks afterwards. As you can see, very early on in the piece, the ethics application goes in and the CTN is submitted. And then while that approval process is underway, we're preparing all of the other documents ready to go. In Australia and New Zealand, we don't need the CTRAs signed with the site in order to do those ethics submissions. So that does give us a little bit of flexibility in that we can have our work order in place with the sponsor and we can get that CTRA signed as close as a couple of days before the site activation. The investigational product can be shipped after site activation, as long as we know that it's going to be available and on its way pretty much afterwards. Then most of our sites are happy to start screening. In New Zealand, as you will see, it's an almost identical startup process. The key difference in Australia versus New Zealand is that Australia, you just do an ethics submission and the ethics committee worries about getting the scientific review. In New Zealand, the submission is done to the 2 committees in parallel. So you submit to the ethics committee, they take care of the ethical aspects of the trial. You submit to the Scott committee and they're reviewing the scientific aspects of the trial. As I mentioned earlier, that sometimes can be an advantage because you do have a dedicated scientific review committee in New Zealand. It does allow for sometimes a faster review because they're focused on just the scientific aspects of the trial, and they're also very familiar with seeing the phase 1 protocols, particularly the first in man. In New Zealand, the MedSafe approval letter is the trial approval, and that acts as the import license. So in both New Zealand and Australia, we don't need a separate import license in order to bring the investigational product into the country. So if we have a look at the R&D cost incentives and what that means for the total cost of the trial. So if you take US and EU as being the most expensive options for running the trial, and that's based on a number of factors, including currency exchange rates and the cost of labour. Australia, before you take the R&D incentives into play, is about that 30% cheaper. Once you take the R&D rebates into account, that does require you to have an Australian entity. It also does require the staff delivering the trial to be on the ground in Australia, but that can reduce the total cost by as much as 60%. As an example, New Zealand without the rebate scheme can also be up to about 50% cheaper. New Zealand does have a rebate scheme. It's about a 15% tax credit. Similar process to being able to achieve the Australian rebate in that you need to have a local office in New Zealand. It's probably safe to say it's a less established rebate than Australia, but it certainly is something that's available. The choice of New Zealand versus Australia, yes, cost is a big advantage, but there is also other reasons that you might decide on one country over another, and that is primarily around your study design and the ability of the individual sites to execute specific assessments in your study or to have access to a particular patient population. So just to give a little bit more information about the rebate scheme in Australia, which is the one that most people are familiar with. So there's a few things that you need to do in order to achieve that rebate. You must have a local entity set up in Australia. You must have at least 50% of the total value of the work be delivered in Australia. Now, generally speaking, say for a phase 1 trial, your CRO costs may be about $1 million. Your bioanalytical costs will probably be about $500,000, but your site costs will be the most significant portion of that. So by the time you take your BioA, your site, and most of your project management, startup and clinical costs for the CRO, you will more than achieve the 50% threshold required to qualify for the R&D rebates. Do need to have the contract signed between the Australian entities. There is some ability to work around that, but it's really important that the Australian entity is involved. And because you need to hold a local office in Australia, you therefore have the responsibility to be the local sponsor. There have been instances where the IRBs have asked for evidence as to how you're going to meet your GCP obligations of sponsor oversight. So that's certainly something that needs to be taken into account. Normally, if you didn't set up a local office in Australia, your CRO would take on the local sponsorship for you, but that obviously isn't an option if you want to claim the R&D rebate. So importantly, across Australia and New Zealand is our access to patients. So we're a government-run healthcare system here, which means that most major cities in New Zealand and Australia have a couple of key tertiary hospitals. We don't have an insurance-led scheme here, which means that there is a lot of collaboration between the phase 1 units and the hospitals in terms of access to patients. Patients also, as I mentioned earlier, have limited access to some of the groundbreaking medications, and that's because the government has to go through 2 steps. Firstly, they have to approve the medication. Secondly, they need to fund it, and it's often the funding that means that patients don't have access to that medication. So that means there's a high degree of interest in a patient population in participating in a clinical trial, even where it's an early phase and they may not themselves get direct benefit from it. We still find that it's very easy to recruit patients into early phase studies. It means that the collaboration between the phase 1 commercial research units and the hospitals is really key. And that's where the choice of site that you use for any particular study is really important. That you make sure that if you want to move from a healthy volunteer to a patient population, that the site that you're partnering with does have that key ability to recruit those patients. There are 2 types of sites that we regularly work with in Australia and New Zealand for early phase studies. So the group that we have on the screen here now are the domiciling units. There are some other units that do have domiciling beds, but they're relatively new, and so at this point probably aren't the best choice, particularly for a first-in-man study. So the ones that we have here are a well-established network of sites. These guys have been around for a number of years, have a huge amount of experience. Importantly, each of them is either co-located or within a major tertiary hospital. So they do have that collaboration already in place that's really critical for both the access to patients and the access to the specialist assessments that we often use. Touching on investigational products. So as I mentioned earlier, in New Zealand, once you receive your MedSafe approval letter that acts as your import license, in Australia, once the TGA notification process has been completed, that also allows you to bring the IP into New Zealand. We don't need to go through a separate import permit process. The only time that there is an extra step that we need to consider is if your investigational product is being manufactured in China and it's the first time that you're exporting the product from China, there is a process that we need to go through, and that's around the availability of what China classes as the regulatory approval. But it's the availability of that MedSafe approval or TGA notification. It doesn't add a huge amount of time to the process, but it is something that you just need to be mindful of. It does add a week or 2 to the overall startup timelines. With all of the sites that we work with, particularly in the first-in-man space, they all have pharmacy capabilities. What's really critical is that across New Zealand and Australia, per clinical trial legislation, we don't need GMP certification of our clinical trial sites. So they are able to do quite a significant amount of compounding themselves. There is also the ability to use external vendors if there is a step that goes beyond the capability of the site. In terms of the IP itself, we don't need GMP certification. If your product has been manufactured in a GMP facility, then certification of that facility certainly helps. Otherwise, all you need is a certificate of analysis and stability sufficient for the duration of dosing. So what that means is if you're starting with a single ascending dose that has just 1 day of dosing, then 1 month of stability is more than sufficient for the initial submission, and you can continue to generate stability data throughout the life cycle of the project. In New Zealand, it is a submitted document. In Australia, it's not submitted, but it does need to be in hand by the time the site is activated. If we need to, we can ship IP under quarantine if there are some final manufacturing steps that are required, but otherwise any of the sites in New Zealand can receive, or New Zealand and Australia can receive that investigational product, even if it's ahead of site activation. It's also very easy to bring in comparator product or any product that may not be approved in either New Zealand or Australia. We simply add that to the clinical trial application as an investigational product that allows you to bring in any medications from anywhere in the world. So this is particularly advantageous for things like biosimilar projects, or if you are wanting to use a comparator or a particular brand that is not available in Australia and New Zealand, that's no problem at all. So if we look at the overall timelines of running your first-in-man trial, you can see that the Australia and New Zealand clinical conduct is quite a lot shorter and is able to finish earlier than other key jurisdictions. Because in the US, you need that IND process, maybe not the pre-IND, but certainly the IND. It does delay the point at which you can enter the clinic in the US. Just for comparison, for those companies that are looking at China for their development, if you were to run your first-in-man in China, it's actually the slowest into clinic. For those of you considering China as part of your later phase development, there is absolutely a way that we can expedite involving China in the phase 1 trial so that you have met the required steps in order to be able to add China to your phase 2 and 3 development. That can be really important for the later phases of your development to make sure that you don't have to run an individual development program in China. And obviously with the large number of patients that are there, it can be advantageous to the conduct of your overall development program. So that's certainly something that we're more than happy to have a chat with you about and let you know about what solutions there are available there. Okay, really importantly across Australia and New Zealand, and this is linked to the lack of an IND, is the development pathway that you can follow. So a traditional development plan might be doing a healthy volunteer single ascending dose, then doing a multiple ascending dose either in a separate or combined protocol, and then having to go to a single indication phase 2. And that's because obviously in the US, your INDs are filed by indication. In Australia and New Zealand, the approval is granted per protocol, not per indication. So what that means is that we can take single protocols that might start with a single ascending dose in a healthy volunteer. Within that same protocol, we can pivot to either healthy volunteers or patients for the multiple ascending dose. Whether that's a single site or needing to go to multiple sites doesn't matter. We're able to do that in the same protocol. Then most excitingly, if you have an asset that you have multiple indications and you're unsure as to which one you want to take as your lead indication, we can create a protocol that has multiple proof of concept arms. You can trial each one of those in the individual group of patients, and then you can make a decision as to which one you take forward as your key indication to open your IND. So it does allow you a look-see much faster than you're able to get in the US because you would have to file separate INDs. So then taking your first-in-man trial, so you've taken all of the advantages of the startup timeframe and the cost advantage, and then where next? Obviously, New Zealand and Australia not having large populations, great for early phase, but they're unlikely, bar a few indications, to be able to complete a phase 2 or a phase 3 study solely in region. So what we have a lot of experience with is where you start with the phase 1 study in Australia and New Zealand, and while that's ongoing, you're preparing your regulatory applications in either the US or EU. With the human data from the single ascending dose arm of your first-in-man study, it can often help to expedite that regulatory approval. What that then means is either within the same protocol, we can expand across and add extra regions for the 1B or the phase 2 component of a single protocol, or as soon as the results from the phase 1 are available, you're able to move straight into the phase 2 because your regulatory application is already there. Because the Australia and New Zealand population are considered homogeneous with the US and Europe, the data generated from ANZ is very well accepted by the regulators. So there's no issue in transitioning out of region for later phase studies. So we just have a couple of case studies and success stories that we've run here in Australia and New Zealand. So obviously in the oncology space, there's a number of companies that have been able to run their first in human via the CTN pathway in Australia, getting that really fast startup timeline by using the private sites in Australia, and obviously, as we've mentioned, no IND, significant cost savings, and being able to get into that patient population as fast as possible. So that trial moved into the expansion faster than it would have if it had started in the US. Second case study that we have here is an autoimmune study. So this was a first in human. We were able to do the startup in less than 2 months, which is pretty standard. Opened the IND in parallel with the phase 1. So particularly for this autoimmune, this was really important because this is quite a competitive space at the moment, and access to patients in New Zealand and Australia is very competitive. So we wanted to make sure that we were moving out to the US as fast as possible so that we were broadening the patient population that was available. What it meant was as soon as we'd finished the healthy volunteers, we were able to start the recruitment of the patients less than a month after that had completed because the IND had been opened in parallel. What that meant was that the sponsor was able to take advantage of the cost and the faster time to clinic, but they were able to keep their strong team that had started with that project, added in additional CRO team members to be able to support the expansion into the US, and it was a seamless transition. So this gave them a much faster pathway to the patient signal that was really important for the later phase development decisions. Okay, so how do you ensure a successful development pathway? So what's really critical is that there is no such thing as too early in terms of partnering with your CRO. We really like to speak to clients and sponsors 12, 18 months before they're considering going into clinic, because it allows us the time to work through all of the downstream implications of running the trial in Australia and New Zealand. And by that, I mean, it's really important that you consider your end market and then work backwards to make sure that your phase 1 study has all of the components in it that mean that you won't have to do additional early phase studies later down the track because you forgot to include China, for example, or you have an oral compound and you didn't think to put a food effects study in the same protocol, and so that means you've got to run a separate study. So the earlier that we can get in and partner with you, the better that we can help make those decisions and ensure that everything that might be rate limiting is dealt with ahead of time. It's really important that you choose a CRO partner that is more than just a set of hands. It's more than just a delivery solution. It's making sure that you're getting that strategy and that ability to seamlessly transition from ANZ out to other regions within the same team, because the relationship that you develop with a CRO partner is really critical, and it's certainly something that you don't want to have to restart with each clinical trial that you're undertaking. All right, so just finally in conclusion, so Australia and New Zealand, we've got the speed, the cost saving and the scientific rigor. So the threshold for the data that you need in Australia and New Zealand to get in your first in man is no lower than anywhere else in the world. It's just less red tape and less paperwork to get into clinic. We've got the easy access to patients with multi-indication capabilities that allows you faster decision making of your overall development plan. We've got the R&D rebate available in Australia, which gives you that significant cost saving. In New Zealand, flexible ethics and the Scott Committee, plus really good access to patients allows for scientific innovation. Combining Australia and New Zealand within Asia or US and EU will accelerate your overall development timeline and de-risk the scale up. And then obviously partnering with Worldwide makes sense because we're able to give you that strategic support early on in the piece so that your transition from phase 1 to late phase is as seamless as possible.
Thanks, everybody. This webinar will be recorded and I believe sent out. So if any questions do arise for Kate, you can certainly reply once you receive that link, and we'll make sure that we get back in touch with you to answer any questions.